Ming, let me start with you. We saw you yesterday talking about some of these novel therapies. So how has you've presumably been practicing through this shift into precision therapy? How has it changed your practice? What do you
I think, to me, the most exciting thing is ctDNA. It's a sort of new technology, unlike tissue biopsy, where you got problem with the biopsy cell and compared to ctDNA, which is a dynamic assessment of what happens.
That's circulating tumor.
Yeah, circulating. Yes.
Take it from
ctDNA.
Yeah. Okay.
So I think it's going to be transform our practice over the next decade, just like mRNA I'm speaking about. So I can speak from my experience from trials I've been doing. So I did this FLORA trial looking at osimertinib, which is amazing drugs. You cure a lot of patients with EGFR mutation with the ADARA study. So what we found in ADARA in our FLORA study is that when you look at ctDNA pre-biopsy and track them longitudinally, we can see they actually precede radiological progression by about 3 to 4 months. That's amazing. And not only that, technology is improving all the time. So now we got NGS as well. Before we use a PCR test, and we can look at the resistant marker, and then we can tailor treatment, and so we can track real time this cancer. So that's sort of metastatic setting. Now, in terms of the curative setting, we call it a ADARA study. So this is a patient who have operable cancer, the EGFR mutation, and you put them on osimertinib, and you see this amazing survival benefit, 5 years. But when you look at ctDNA level, if they have what we call a minimal residual disease lab on the ctDNA, they're more likely to predict relapse. And now it sort of brings back to this issue about health economic, because as you heard yesterday, osimertinib, 1-year treatment, I don't know how much it costs, probably a lot of money. And then applies to also immunotherapy, 1-year treatment, £100,000 or $100,000. So it sort of changed the landscape now. So now we're going to what we call the non-oncogenic cancer. So we're talking about neoadjuvant treatment for lung cancer, where they give chemo, they give immunotherapy. And what we are seeing is that they all got ctDNA present at the baseline. Now, if you completely disappear after neoadjuvant treatment, most of them don't relapse. So now we are coming to this era where we are talking about what happens if you're ctDNA negative. Should we stop treatment? Should we stop adjuvant immunotherapy treatment? So we don't avoid the toxicity, we avoid the health costs, we avoid £100,000 or $100,000 spending. And we need to focus on ctDNA positive patients. So trials are ongoing to that. So look at ctDNA positive, what happened to them? And can we actually escalate or de-escalate our immunotherapy treatment? So this is sort of a new frontier now in lung cancer research. So we're going to hear this story for the next sort of decade or so. So it's a new technology. So to summarize, ctDNA liquid biopsy testing is not like the tissue biopsy anymore. It's sort of new era. We not only track tissue biopsy, one snapshot, you get a result, you forget about it. But with liquid biopsy, you track them, you monitor them, evolution, new marker, new mutation, we can design new treatment. Right. And or even we track them closely, PET CT scan, they relapse, we can give SABRE treatment, localized treatment. So we avoid all this toxic treatment long term. So a lot of cost saving for our healthcare system.
And relatively, I mean, it's non-invasive for the patient.
Right,
well.
yeah.
So, right, okay. So you've basically got a dynamic signal that you can track someone over time by essentially taking blood samples and see their response to treatment and tailor the treatment as they go through the cancer process.
Correct, yeah.
Right, okay. So we're seeing these great advances in these genomically targeted and genomically modified treatments.
Yeah.
But I want to extend, because that's often how we conceive, quite often people conceive of precision therapy as being centered around genomics. But I know probably Michael and Nina are going to expand that theme beyond simply genomics. And so, Michael, you first. So how would you see precision?
Yeah, for me, what you described, targeting a disease, is one pillar of precision medicine. Of course, when I practice, I ask my patient first, what do you actually want? And then the next, I think, I try to describe the patient better, not just the target. So EGFR is an interesting target. You described it. It's an oncogene. It's fairly straightforward. However, as you said, most of the tumors are probably driven by oncogenes we don't yet know, or a combination thereof. And therefore, I think it is very important that we use diagnostics at the micro level to really describe the patient well. If I want to give immune therapy, is the patient actually capable to sustain a bispecific antibody, or is there no immune system worth trying a bispecific antibody? Should I give something like CAR T cells, where I have the options between immune interventions? I think that is really where precision medicine comes to play, is to really better describe the patient. And it can be very simple. It can also be, we use a lot of advanced analytics to describe patients better from their health status, from their ability to tolerate treatments. We have a number of very interesting antibody drug conjugates in our own pipeline. And of course, these are interesting medicines if given to the right patients. However, given to the wrong patient, they cause a lot of damage. And that's where I see precision medicine coming in more and more and more. Today, so we test the tumor target, as you described, EGFR. Tomorrow, I think diagnostic needs to be much, much broader. It needs to be not following the treatment. It needs to be following the patient. It needs to describe the patient to really, I think, bring individualized medicine. So I don't know whether I will see this in my clinical practice, but I'm hopeful.
I can speak from my experience with TracerX. So this is a longitudinal study in the lab by Charlie Swanton. So we biopsy them. They relapse, we biopsy them. We track them with our blood tests. I think I was helping Chris Abosch on the ctDNA analysis. So we can look at the phylogenetic tree. So this genomic evolution that we know human project costs over 2 billion. But now you can do a genomic testing for less than £1,000, even £500. So we track them and see what happens with this cancer, the evolution going on. So we can see the clonal one present in all the cells, the subclonal one, and we can track, subclonal one emerging as a primary driver of disease progression. And by looking at that subclonal, I think in the future we can sort of work out what kinds of mutation or what kind of acquisition. If they have sort of marker we can treat, like HER2 and all that, we can give a specific drug. So I think very exciting. So it sort of helps us in mRNA technology as well. So looking at all these mutation changes, evolution profiling, real time is quite amazing for medicine. So this is where we are going in the next decade or so.
I think it helps tremendously to guide patients or to guide treatment for patients once you have a diagnosis. And of course, once you've sequenced the tumor, I think otherwise ctDNA is relatively insensitive, I guess. And in diseases where we don't have the tumor to sequence, I think it's not there yet. It will be probably there tomorrow, but it is not there yet unless I can sequence the tumor and make sure that I can truly follow and see what I want to see. But I think it's a really nice technology. I think it's one part of moving forward in the world of diagnostics.
Okay, so I am not a scientist, I'm a medic, so I'm going to try and summarize, before I come to you, Nina, to expand this precision idea out. I'm going to try and summarize for myself, and you can tell me if I'm wrong. So step one is, well, cancer is a disease of the genome, so you get genetic mutations. So the first stage of precision therapy is there's a gene that's broken, you spot it, and then you have a targeted therapy that might attach to that. I'm being simplistic, but bear with me. Then, I guess though that it's not as simple as that, because that there could be other broken genes, or there could be other correlates with that, because the genome is very complicated. And so we're learning that it's not just a one-to-one mapping, but there may be other features of the genome that we need to map both over time in one patient, but across patients as well, that might help us target therapy. So you go from a one-to-one mapping of target to therapy to and this is where AI and so on may prove useful, to a pattern recognition which may change over time, which helps you to select, give a prognosis and direct your treatment. But then beyond the genome, there are lots of other markers that we may build into our thinking around this from the proteins, the phenotypic expression of those proteins, patient preferences, and that brings me to Nina. So if we're really going to be precise and tailored about our treatment, we need to build in all those bits of information as well that go beyond simply the biological markers. So is that something you would?
And actually, biological markers are very much involved. And I'm happy to say that tomorrow's medicine is today, because biomarkers in my patients what I track every day. And actually looking at that broader picture that you described is somebody's immune status, looking at the broader picture of the person's performance status. And more importantly than just biomarker patterns is how do we actively modify them? So looking at immunotherapy, for example, that's a pan-tumor indication for multiple solid tumors with immune checkpoint inhibitors. We know that patients' baseline inflammatory status matters. So if your neutrophil to lymphocyte ratio is up, if your CRP at baseline is up, if multiple combined prognostic markers such as LIPI as an index or SII are all in the wrong direction, the patient is inflamed at baseline, immunotherapy is not going to land as well.
Right.
So it will actually, you won't get such a good response. You won't get the right kinetics of a specific targeted tumor response. You'll get this non-specific inflammation, potentially some more IRAs as well. So if we have expensive drugs, we really owe it to our patients and the health economy to optimize patients. So that's something that the current pure medical toolkit does not have, but the integrative oncology does have. I can change my patients' CRP and inflammatory status at baseline, but modify their nutrition, lifestyle, and doing other things, including looking at the gut microbiome and the gut immune axis. I'll give you an example. One of our patients with high-grade serous ovarian cancer had tried low-dose immunotherapy before in another country, came to the UK, and oncology decided to re-challenge and try pembrolizumab and lenvatinib just to see in this recurrence whether it would work. She had colitis as her last complication of treatment. Her baseline CRP was 8.4. Okay, that's significantly elevated from my perspective, and LR was elevated. We knew she was heading to pembro, and the potential risk was getting another colitis and just completely ruining that line of treatment. By actually providing an integrative oncology intervention in 3 weeks, that dropped from 8.4 to 0.8.
What was the intervention?
Yeah, so it's a multimodal intervention. Okay. Honestly, I could just describe it, but it will take us most of it. But multimodal integrative oncology, which includes nutrition, lifestyle, optimizing vitamin D status, other things that are relevant. But you could drop it to 0.8, you normalize the NLR. This patient has now been on this combination of treatment for 9 months. There's no colitis and there's no evidence of disease. Okay, in high-grade serous ovarian cancer with this treatment, if there's any gynae oncologists here, that's pretty unheard of. So the key thing I think for me to look at is when we're looking at things like immunotherapy or other targeted therapies, whether it's TKIs, etc., what are the predictive markers of response in terms of baseline patient states, not just tumors, because otherwise we're at risk of that picture, right? Of sitting there looking at the tumor, forgetting the patient. But putting the patient into that ecosystem, what are the patient factors? Is it baseline inflammation? Is it metabolic health? Is it their gut microbiome? Which we know there's boatloads of data on the gut microbiome and some keystone species influences on immune checkpoint inhibitor treatment. And then not just saying, oh, well, you're now in poor prognosis bracket, but saying, let's reclassify you. Let's actually do an active intervention to change those biomarkers because they're modifiable. I can't modify your EGFR. Whatever the exon mutation is, it is what it is. It will evolve past it at some point, but that's fine. But I can absolutely modify patient-related markers to help support that line of treatment better. And that's the bit that we need to look at.
I can come back to that issue. I think I sort of jumped the gun talking about ctDNA. I think lung cancer is pretty lucky because we got this discovery of what we call oncogenic-driven cancer. So you got EGFR, and you got ALK, you got BRAF, you got so on, skipping mutation. So when that sort of came around about a decade and a half ago, there was this issue about testing, turnaround time. And, patient cannot wait stage 4, 3 weeks for the results. A lot of centers don't even test it. So we need, in the West, we're probably lucky. I think we're cutting the time now. So we actually use ctDNA to actually turn around time within 24 hours to EGFR, the common one, easy to test PCR. But this is sort of issue, we sort of now have to try to work out for the third world, to make sure they all got tested, all got the right personalized treatment, and how to shorten the turnaround time. That kind of issue, which is sort of a broader healthcare problem for the undeveloped world that we take for granted. So that kind of issue, even I think in the West at the moment, turnaround time to look for all that kind of mutation, Alvira, and tissue, 3 weeks. Hopefully, with liquid biopsy, we can turn it 10 days time because we won't crack on with the treatment.
Right.
So sort of issue, sort of neglected in a way because I'm sort of focused on ctDNA story, dynamic testing, real time, no longer science fiction, but tracking, save money, and all that kind of consequence in the next decade or so.
And it brings up a couple of critical questions, but just before I get to those questions, how much of a game changer is this advent of precision biomarker usage across, from how much is it impacting patients' lives?
Well,
Huge. I mean, metastatic melanoma before ICIs, right? Death sentence, right? Now we have 5-year OS at a very reasonable state.
Okay.
So it is a complete game changer when we're looking at actually molecularly targeted therapies, and pan-tumor indications were never a thing, right? Before we had things like looking at properly at DMMR and dynamically looking at MSI high, etc. So when we're looking at biomarkers now, the fact that there never was a drug that could be taken in a pan-tumor indication before that, I don't know of any.
I think we're really good at targeting the tumor. I think we have really evolved, and in diseases like melanoma, which is an immunogenic tumor, which we knew for a long time, and certain lung cancers, we really succeed. However, you described it quite nicely. We are less well in describing the patient, which patient should get the treatment. What you described is nice, this total change, and I'm a believer of it. But however, measuring it is incredibly difficult. To go to my payer also to say, I want this kind of treatment because it changes this biomarker. It doesn't work. I mean, things like microbiome. We know it works because we can transplant the microbiome and get someone who is actually not responding to immunotherapy to get them to respond. However, how do we measure that? We have no clue. And to make this into a real interesting therapeutic modality.
Into a standardized.
We are not there yet.
Yeah. So that was my question. That one of the complexities it raises is it moves so fast. The field is so dynamic, and you're kind of selecting patients. How do you? The traditional model is you have an intervention, you perform an RCT, and then you get it into the system. But here you're having to You're having to develop in real time different models and work through patients, and it's how do systems adapt? How do you adapt that, or how do you make that fit into a system of cancer care when it's so dynamic?
For me, at the moment, we don't adapt it very much. I mean, we look traditionally at the patient now, and we look at the tumor, we target the tumor. We don't increase our spectrum of diagnostic possibilities because it came up earlier. Diagnostics cost money. We do diagnostics when we have a treatment for it. That's the way we grew up in medical school. It's we will do that and no more. So we haven't changed our glasses yet, but we probably need to change our glasses in the future if we want to really do true precision medicine. Because also when you use a simple intervention like AI, if I look at something, this is for a hematological cancer, and I look at it, over years, I wouldn't have seen that a particular marker, which is a very simple marker, comes out of the Coulter counter every day, is really one of the markers influencing whether or not a patient will fail their line of treatment. We need really to look unbiased from what we've learned and how we looked at patients in the past. And for that, I like AI. I'm a fan, but I also see its limitations and criticism. But for its unbiasedness, I really like AI.
I think the thing about AI is what we need to feed it is the right thing, right? The models you train it on is really, really important. And I think that when we train it on data that's purely tumor driven, we're missing the patient part of the picture. So I think if we're going to go to precision medicine, not just actually precision oncology tumor therapy, but actually treating the patient, then we do need to make sure that when we feed and train those models, it incorporates patient-related biomarker data, as well as other baseline data, the general stuff in terms of demographics and other things. Because there is definitely a healthcare inequality thing, and there's also genomic differences in responses to different therapies, which we know is completely unexplored. And actually, our trials are really terrible at exploring it because we're not recruiting the right people to the right trials.
Yeah.
So I think we really need to, when we're looking at AI and maybe complex prediction models, to make precision medicine the reality that will take our, PD-L1 status and EGFR mutations and everything else into account, but will also look at those baseline inflammatory markers, those course of antibiotics and other bits and pieces.
Yeah.
To do that, we need to feed it good data, and good deep data is actually quite hard to find, I would say, at the moment.
Look at, this dynamic liquid biopsy story. I think it's very exciting. So you can imagine, you track them real time and you identify something with mRNA technology. You can develop vaccine against it before it become radiological progression.
Yeah.
I think this is sort of the future. I hope it's no longer science fiction, but that's what I hope it will happen. Yeah, lung cancer especially. So ctDNA, liquid biopsy, confirm, disappear, track them, come back, track them. Okay, we've discovered this thing, mRNA, bang, before radiological progression, or we monitor them more intensively. If they relapse, stable treatment, localized, carry on with the standard treatment like osimertinib. So I hope this is sort of the new world now, and we just have to wait and see. So hopefully, the next generation, we need more young scientists to actually develop this technology.
Any questions from anyone?
Can I just make a slight comment, I guess, mainly on the breast cancer side before we finish up? It's just, taking that into recent ASCO data on the breast cancer side. That's exactly what they tried to do with breast cancer. They looked at ESR1 mutations, which confers resistance to aromatase inhibitors in breast cancer. And they applied a new drug called camizestrant to see if it actually, before you get progression based just on mutation, if we switch treatment early, could we affect outcomes? And one of the big criticism was that they haven't demonstrated OS yet. They haven't demonstrated overall survival benefit. So part of what we do with this dynamic adaptive strategies is to get them reimbursed and get them into practice, we still need to demonstrate conventional oncological marker impact. So that's another thing, is like we need to know whether it actually makes the final difference for us to be able to make it a proper reality.
Which is fair enough.
Maybe follow up on the Serena study, which you referred to. I mean, I think everybody accepts that the data is really very interesting. Yeah. The problem, of course, how do you implement it? I think you need to screen with the current technology approximately 10 times more patients than you end up actually switching the treatment. And then, yes, there is, of course, a lead time bias in it, which we will have to see whether it does result in overall survival.
Right.
But it, of course, I completely agree. It is the future where you have a marker to look at ctDNA. However, in clinical practice, outside studies, it is very difficult to screen 10 times more patients than you end up.
Yeah, I agree. I haven't covered the standardization, the commercialization, the reimbursement, the teaching, or the new clinician, all this new data. So I think this is beyond my expertise.
Yeah, so that's the challenge that I wanted to try and address. But let's have a question from the floor. Can't quite hear you. Can we switch the mic?
I practice in the US. I ask for liquid biopsy for all my patients. The problem in the US is basically some insurance will pay and other insurance will not pay for the liquid biopsy. And the reason they said will not pay for liquid biopsy is because it's considered experimental. It's within the standard of care. So how do you convince in the UK the insurance to pay for liquid biopsy? Because we know it will be the future, but the problem is the insurance does not pay. The patient have to pay out of their own pocket. $1,000 is a lot of money.
Yeah, I mean, in the UK, well, we have a a directory of genomic tests, which is nationally set, and we also have clinical guidelines, which are nationally sort of approved through an organization called NICE. So that's how you would get these things that's the standard procedure for getting any new technology into the system, as it were.
And private reimbursement varies in the UK.
Private,
for liquid biopsies. And I think we're still in I think some of the paradigm shifts need to change in our thinking. There are two things. One is that real-time monitoring needs to be a thing. It's like, it's not a one-off test. And we're still trying to look at reimbursement models that are one-off tests. I'm like, that's not the point. The point is I need real-time, over-time assessments so we can design the treatment pathway. The other thing I think that does need to change is that I still see all tissue samples being sent for genomic analysis at progressions 2 years later. We're not going to trust the potassium from 2 years later, but we're perfectly capable of sending tissue to try and see what my next line of treatment is. After 3 lines of treatment, your selective pressures, like what you're describing, your subclones, are completely different. So we need to really, I think, change the thinking around this new technology.
Maybe also to come back to what you say, I think experimental setup, I think ctDNA will for a long time be an experimental setup. What we have to change in our mindset is we treat clinical trials, the whole of precision medicine, individualization of treatment will be for a long time experimental. And we have to do continuous clinical trials. And the clinical trial, the journey becomes the target, not the outcome of the clinical trial. And I think that's what we have to change in our approach to precision medicine. The journey becomes important, not just the outcome. If we look at this, we get the same responses as you get. It's an experimental treatment because we don't have the hard evidence, the overall survival or whatever. However, if we do continuous clinical trials, and there are some, we can really truly advance precision medicine.
I think it's important because we are now in this new era, not like chemo, 100 pounds kind of era. We're talking about one shot, 5,000 pounds, 10,000 pounds, that kind of shot. So 1 year, 100,000. So all this testing, is nothing compared to the real cost of medicine. And I think this Human Genome Project, 3 billion initially, now $500, 500 pounds, So I hope the costs keep on coming down, and we can sort of save a lot of money long term.
I mean, I suppose there's an analogy. The difficulty, I suppose, is that when you say liquid biopsy, it's not just the liquid biopsy that one has to reimburse for. It's what you do with that biopsy, what tests you apply to it, and what predictive algorithms do you sit on top of those tests, and then how do you respond to that? And so I guess that's one of the challenges with this, isn't it? It's that it isn't one thing. It's a whole range of things that you can do with a sample of tissues and a sample of data from a patient.
Usually that's built into the pipeline, though, isn't it? Like that sort of decision making that you're talking about in terms of the reporting that comes out, when you're doing this in a commercial, not a research level, but at a commercial level, that's built in. But I think in terms of the potential for, and not just ctDNA, but other liquid biopsies to really transform patient care is huge, So we've had things that were BRCA1 negative on NHS testing came back as BRCA positive because the technology wasn't so good in time when the patient was originally tested, that just opened up another line of treatment for somebody because we did a liquid biopsy that also looked at some of their germline genetics. So I think there's a lot of potential here. I think, answering that question, I think it is all experimental right now, and how we move forward in reimbursing it in the right way so patients don't miss out on lines of treatment they're eligible for is important.
So so sticking our neck out here, what would be one way of doing it would be to be hyper vigilant about regulation and saying, and the procedure say, you must take the biopsy at day 1, and then you must, then you can run these tests on it, and if test this, if that, then, and to regulate the entire algorithm and reimburse alongside. The other way is to kind of completely deregulate. So whatever outcome you get, great, we'll evaluate it and we'll pay you a sum. And so what is the right way when this is such a dynamic, and I don't, this is an impossible question maybe for this panel to answer, but given this complexity, what is the right way? How can we enable the system to operate in a way that's safe for patients, doesn't allow cowboy behavior, and at the same time...
I mean, we see that in cancer. We see the drug-like trials which are existing. I think there are about 10, 15 in Europe running. So these are studies where patients are screened for all kinds. It's still limited to the tumor rather than the patient, but at least it's a progress that patients are screened for all kinds of mutations, and where there is a licensed treatment, not licensed for that particular disease, but effectively targeting that mutation or the protein expressed through that mutation. That has been made available. And these studies are collaboration between government and academia and industry. So industry gives the drug, government gives the funding, academia gives the intellectual prowess of it. And I think these are really good co-creations of a way forward, at least where tumor targeting is concerned. We should do more of them. I think they're very popular in the mid-sized countries. They're not yet so much taken up. UK has Reliance. I think you have one study of the drug-like type.
I run many trials. One of the problems is commercial study. You're not interested in escalation, desescalation. They're more interested in escalating.
Yeah.
And I think trials, we should tell the pharma company, you do this trial, you fix it, say the cost factor, and then give a overall view, how much is actually, 2 years, 3 years, 5 years duration, how much the cost actually for the patient. So I think it sort of put pressure on them, instead of trying to sell more drugs. And we can say, you don't need 1 year treatment, you don't need 2 year treatment, 6 months is good enough, so your ctDNA disappear. Patient, no toxicity, cost saving, everybody benefits. So we can focus treatment on the right population. So maybe we should start thinking along that line as a trialist. But we're not doing that anymore.
Yeah, because that replicates what would happen in the real world.
Correct.
Yeah.
Well, there is a group called the Cancer Medicine Forum, which is an EMA body, which looks exactly that. So for my friends, I'm part of that. So we are looking exactly at that. How do you optimize treatment, as you described, driven by biomarkers, driven by other insights? And yes, it's very difficult because, as you say, we can do it like cowboys. Everybody sort of does their own scheme. I finish here, I finish here, I finish there. Or we try to do studies. But studies will take, of course, a long time. And I believe really need cooperation. They cannot be done by industry. They cannot easily be done by academia. We see this now. We're trying to pull off a prostate cancer study with the EORTC. That as a Cancer Medicine Forum, that's very difficult for rules and regulations.
I think, I guess, the answer to your question is a balance, really, is like you said, you can't do either one or the other, right? Platform trials, of course, a great way of looking at different ways of doing precision medicine. But I think the idea is actually what we need to do is be able to make a kind of flexible algorithm, really, that actually will be able to individualize therapy for patients. I don't think we're there yet in any way, shape, or form. So that's why it's so difficult for us to position the liquid biopsies for everybody, because we don't have unlimited funds, and we really need to be careful of that. But I think the answer is somewhere in the middle, right? That's the idea. How do we get there is probably going to take us quite some time to figure out. And it might be, the progress will be different, I would say, across different cancers as well, not forgetting the fact that actually it'll probably be a lot trickier to do unlike things like, lung cancer, for example. It might be a lot trickier to do in places like ovarian cancer. So we need to probably also specify it per actually cancer type as well, because we have a lot more research to be able to guide that algorithm in certain cancer types.
But of course, it may not be an algorithm if AI is involved.
Exactly.
Today's AI.
The dynamic decision-making AI box, shall we say?
So that makes it even more complicated.
Exactly.
Yeah, but it's not. It's a problem with medicines always, because we are all intelligences as doctors, and we behave slightly differently from each other. So it's not a problem that hasn't occurred before in that sense, that we learn from our practice and we practice differently tomorrow than we have yet.
It needs to be a living guideline that is dynamically adjusted, like platform trials drops their arms, for example, and adjust their protocols as they move forward. It's the same thing needs to happen. It's actually when we say, you know what, I don't need ctDNA every 6 months, I need it annually, and that will be good enough. And yes, if you're ctDNA negative, I then don't need to change you, can stop your treatment. All of that will need to happen at some point, but we need good data. And I would still suggest definitely incorporating patient-driven biomarkers in that data.
Okay, great. I think we'll stop there. I'm not sure we've solved anything, but it's really, I mean, but health systems need to deal with this because this is the reality, isn't it? So, okay. I think it's networking lunch. People are probably hungry, aren't they? Okay. All right. Okay. Thanks.
Thank you very much.
Thank you. Thanks so much.